This video is on the history of early Earth, some of the experiments associated with early Earth, and conserved processes. So if we look down here, okay, at this graph, you can see on the x-axis is billions of years ago, and on the y-axis is atmospheric oxygen. So a lot of times during this video, I'm going to talk about free oxygen. When I talk about free oxygen, I'm talking about O2. Okay, in water, there is an oxygen atom in water, but it's not free. So free oxygen, I'm talking about O2. Okay, for free oxygen. Write that down. So when we look at this timeline, okay, this line shows the change in free oxygen over time. Okay, so at the beginning of this graph, about 4.5 billion years ago is when Earth was formed. Scientists see protocells, evidence of protocells, which means like cells with membranes, but maybe not all the things we associate with cells, like DNA or ribosomes or anything like that. So scientists see evidence of protocells about 3.8 billion years ago. Protocells are the first structures with membranes. So when scientists first started to think about like the evolution of organisms on Earth, they looked at the conditions of early Earth. And many scientists wondered, how could life emerge from non-life amid the conditions of early Earth? So one of the first groups of scientists to do an experiment on this are Miller and Urey. Okay, so the Miller and Urey experiment was an experiment to model the atmosphere of early Earth to see if they could get components of living structures from this atmosphere. So they looked at the conditions of early Earth, primarily based on, evidence from layers of rock. Okay, and when they looked at this, they knew that early Earth was warm. Okay, and had water vapor, so had water in the atmosphere. So they connected all these chambers. The first chamber had boiling hot water to create water vapor, right? And this water vapor traveled up this tube. Okay, and this was the atmosphere in early Earth. Okay, early Earth had a bunch of inorganic ingredients. Okay, so all of these are inorganic here. We have methane and ammonia and hydrogen gas and water. Again, no free oxygen. There's oxygen within other molecules, but no free oxygen. So they created this atmosphere like early Earth with all the chemicals in early Earth. Okay, and they added electric sparks to simulate a lightning storm, because early Earth had a lot of lightning, right? And they also made sure that this environment contained the inorganic materials from early Earth, which basically means they set up a reducing environment. Okay, which means molecules could gain electrons because there was no free oxygen. So they have the heat, they have the mixing of the chemicals, they have no free oxygen, they have electricity, and that electricity allowed them to spark chemical reactions, right? Then they started to make cool water go around the outside of this container. So they had a condenser that took all the molecules that were forming here based on early Earth conditions, and they cooled them down, because I knew Earth cooled down a little bit. And then after a couple days, they came down here and they collected the condensation. And what they found was they found organic molecules. Okay, so this provides evidence that organic molecules can be made from inorganic molecules. So like short carbon-hydrogen chains, short maybe like precursors of amino acids, that kind of thing. All right. So the Miller and Urey experiment did not prove that cells could be created from non-life, but it did prove that the building blocks of cells, carbohydrates, lipids, amino acids for proteins, basically the monomers of those organic compounds could be created based on early Earth's atmosphere. So that was an interesting experiment. So let's come back up to this timeline. If we notice on early Earth, there's no free oxygen, right? That's why we see this graph at 0 here. There's essentially no free oxygen. We have this reducing atmosphere. This reducing atmosphere allowed for the components of structures that could make up membranes. So we have our first protocells. Scientists think about 3.8 billion years ago. Okay. Then there's chemical evidence in the fossil record there's chemical evidence and fossil evidence of anaerobic prokaryotes at about 3.5 billion years ago, a little before 3.5 billion years ago, emerged the first anaerobic prokaryotes. So these are prokaryotes, one-celled, no nucleus, and they do not like oxygen, which makes sense because there's no atmospheric oxygen, they can survive. Okay, so we have our anaerobic prokaryotes. Then scientists think somewhere between 3.5 and 3 billion years ago, we had the evolution of glycolysis. One of the reasons scientists think glycolysis evolved so long ago is because it is a highly conserved process that's kept the same across kingdoms and domains. So it makes sense that it evolved early so that all the other organisms that evolved afterwards had that process of glycolysis. Okay, so then scientists are looking and they find that about, I don't know, a little 2.8 billion years ago, 3 to 2.5 billion years ago, we have our first cyanobacteria. And these cyanobacteria do photosynthesis. So they put oxygen into the air. Okay. And you can actually see right after they evolve, as there's more and more of them, okay, you can see this huge rise in atmospheric oxygen. That would have killed off many of these anaerobic prokaryotes unless they lived in areas where they could still live without oxygen, right? And that kind of leaves a space in the ecosystem for other organisms to evolve. And we do see other organisms evolve, okay? Once this oxygen level gets a lot higher, we see aerobic prokaryotes evolve. And the aerobic prokaryotes can do more steps in cellular respiration. They can utilize oxygen. They can live in the presence of oxygen. And as those aerobic prokaryotes evolve, it doesn't mean the cyanobacteria go away, but there's more balance between organisms creating oxygen and organisms using oxygen. So you see the atmospheric oxygen concentration decrease slightly, right? And then at about 1.8 billion years ago, you see the rise of the eukaryotes. And you see the rise of the eukaryotes, okay, after endosymbiosis. So I know we've talked about endosymbiosis before, but just as a quick refresher, okay, endosymbiosis, a woman, Lynn Margulies, came up with this idea. This is the process for how eukaryotic cells evolved. Okay, so the endosymbiotic theory, okay, is you have a large prokaryote, okay, and this large prokaryote started to fold in its cell membrane, because this prokaryote has an external cell membrane, but it started to fold in its external cell membrane, creating kind of the prototype of the ER and this prototype of the nuclear membrane. So now this prokaryote has an external cell membrane, and it's starting to create what we would refer to as the endomembrane system. So the enfolding of the plasma membrane to create the endomembrane system. Remember the ER, the Golgi, etc. Okay, now the other thing that would happen is this large ancestral prokaryote, right, is create this nuclear membrane, maybe the ER, okay, is going to engulf smaller prokaryotes, eat smaller prokaryotes through endosymbiosis. So the cyanobacteria is much smaller. It's engulfed by this prokaryote. And then over here we have this aerobic bacterium. It's engulfed by this prokaryote. And these organelles, well, these cells that used to be their own free-floating prokaryotic cells are engulfed by this ancestral prokaryote, and this is what's created our eukaryote, right? We have our nuclear membrane, our ER, and then these aerobic bacteria become the mitochondria, and these cyanobacteria become the chloroplasts there, right? So this larger prokaryote here engulfed the smaller prokaryotes, right? And then they became the chloroplasts from the cyanobacteria and the mitochondria from the aerobic bacteria. Right, and the cyanobacteria that became the chloroplasts are going to do photosynthesis. The aerobic bacteria that became the mitochondria are going to do cellular respiration. So what provides the evidence for this? Okay, and again, we've talked about this before, but it's a good idea to summarize this. The evidence for the mitochondria and chloroplast having undergone endosymbiosis to go into the cell is they have their own circular DNA, like a prokaryote would, and their own ribosomes, which are smaller, like prokaryotic ribosomes. Okay, they can replicate on their own. They have a double membrane. Remember, one was their original membrane from before they entered, and the other one is the membrane they got through endocytosis, right? And actually, the chloroplast has 3 membranes there, right? And they're about the same size as today's procs. All right. So first eukaryotes evolved about, probably through the process of endosymbiosis, about 2 to 1.8 billion years ago. Okay, but these are still pretty small, relatively small one-celled prokaryotes. Okay, then multicellularity, probably between 1.5 and 1 billion years ago, you have multicellular organisms. And then you get plants about 600 million years ago. You get plants somewhere between 600 million years ago and 500 million years ago. You get animals. So let's say like kind of between here. And then when do they actually colonize land? So before this point, you guys, every single thing that we're talking about happened in water. Okay. When did they actually colonize land? We're looking at the colonization of land, I would say probably about 400 million years ago. So, and you can kind of see after aerobic prokaryotes evolved in eukaryotes, atmospheric oxygen concentration kind of stabilizes. And then once we have plants, atmospheric oxygen concentration rises because multicellular plants are going to produce a lot more oxygen. And then we stabilize again with animals who are using that oxygen. Okay. So this is kind of the timeline of early Earth. Another really important thing to finalize this process is this idea of conserved processes. Again, we've talked about conserved processes before, but they are pretty important. So let's kind of summarize them here. Remember, a conserved process is a process that's kept or is the same across kingdoms and domains. And one of the ones we talked about here is the early evolution of glycolysis. And you see it across many kingdoms and domains. And glycolysis is really important because it's so evolutionary advantageous because you can take things that you eat and then produce these ATP energy storage molecules that can be immediately used, right? Immediate use energy molecules. So you see things that are conserved that are really evolutionarily advantageous, okay? Another conserved process is, and structure actually. So I guess it should say conserved processes / structure. DNA and RNA is genetic material, and then protein synthesis. These are all important conserved structures and processes, right? We know that the genetic material is universal. Using that triplet codon chart is universal to all organisms on Earth. And the code is the same across the domains, right? We have 3 nucleotides coding for 1 amino acid, right? Another conserved process is, it's not conserved across all kingdoms and domains, but another important one is really photosynthesis. Obviously, fungi and animals and some bacteria and some protists don't do photosynthesis, but there are some form is present in plants, protists, cyanobacteria. So it's not conserved across all organisms, but many organisms, right? And cyanobacteria, of course, it occurs on the membrane. And in plants and protists, it occurs in the chloroplast. But it's a good example of a conserved process, right? And I think these are important to note when we're talking about early Earth timeline, because as things evolved, things can evolve more than once. Like the same thing, like flight or streamlined structure can evolve more than once in evolutionary history. But these are some things that evolved and were so evolutionary advantageous that they really stayed in organisms over time. So that's the end of this video. Please watch my next video on domains and kingdoms.